Yeah, thank you so much for having me and all of you for listening today. So I'm happy to make this very informal. So if you have any questions, feel free to use the raise your hand smiley or just throw your question in the chat, or just unmute yourself and interrupt me. That's also cool. I'm gonna go ahead and share my screen. So I kind of wanted to talk about two aspects of my work today. I'm going to be talking about the new virus pipeline and catching a virus. But I also wanted to talk about what it was like for me to go from wet lab biology to a more software engineering role slash bioinformatics role, and talk about kind of what I've seen having been on both sides, and what I've learned about what it means to write software for biologists. So I'm actually going to start with that part of my talk. And a lot of my talk, I'm now at the Broad Institute, but a lot of my talk is going to be about my PhD work in Luebbert's lab at Caltech. And so the PACHTA lab mostly focuses on how do we analyze high-dimensional data like single-cell RNA sequencing data. So in single-cell RNA sequencing, you have a tissue of interest, you cut off a little piece of your tissue, you dissociate the cells, and then you read the RNA molecules in each individual cell. And what you get back is literally a text file with a bunch of A, T, C's, and V's. And from that, we align that to a reference, and from that we try to get an expression profile where for every cell you can see the expression for every gene. So you can see how this data becomes really, really highly dimensional if you have thousands or millions of cells and thousands of genes. So my work mostly focuses on these last two parts, where I'm trying to make sense of this very high dimensional, very large data and make it into something that is biologically interpretable. So single-cell RNA sequencing is a transcriptomics technology. And so as that, it's part of the omics technologies, which have really exploded over the last 3 decades in biological research. And I did want to just give you a little bit of a feel of how big that explosion really was. So in 1997, if you were a subscriber to NCBI's GenBank database, so that's a database of annotated DNA sequences, you would literally get sent home these 7 CDs. And that's the entirety of the GenBank database. This is a picture that was provided to me by Luebbert. Today, GenBank contains 2.5 billion annotated DNA sequences. So with modern compression techniques, we can get that down to less than 4 gigabytes. So assuming like 700 megabytes per CD, and like every CD has a width of 1 centimeter, you would get about the height of the Leaning Tower of Pisa if you wanted to put GenBank on CDs. Another database is the Sequence Read Archive, or SRA. And the SRA was only established in 2012. And the SRA is where you would send your sequencing data. So if you generate a sequencing data set, you would deposit your data in the SRA. So it was only established in 2012, but already since 2012, it has grown to over 30 databases. So that's 42 million CDs if you wanted to put that on CDs. So that's just to give you a little bit of an idea of this explosion of very high dimensional omics data in the biological sciences. And there's a lot of different databases like GenBank, like SRA. Here are some of the more famous ones. Ensemble, UniProt, and NCBI are 3 of the most widely known databases, and they each have their own kind of specialization. So UniProt focuses on protein sequences, and Ensemble hosts a lot of vertebrate and also invertebrate genomes. And when I first started in the PACHTA lab, I very quickly became tired of manually searching all of these databases. So they each have a website with a different web interface. And so one of the very first softwares that I wrote when I joined the lab was GGET. And basically the idea behind GGET was to facilitate access to these different databases and querying information from these databases through one combined interface. And so just to give you an introduction to what GGET is, it's basically a collection of different modules that each does one specific task in bioinformatics in a single line of code. It has since outgrown just querying of databases. So now there's also modules like GGET AlphaFold, for example, where you can just give it an amino acid sequence and it will locally compute a prediction of what that protein might look like. And all of these you can just run on a laptop, so you don't need any special computational requirement. Which brings me to one of the first things that I've learned is that that is very important. Your software has to be able to just kind of run on a laptop if you want it to be widely implemented. Also, one very important aspect that GGET covered is reproducibility. So all of these databases get constantly updated. So you can imagine it's very difficult if you have to manually query information from these databases. You're trying not to make mistakes. You have to copy paste gene names, so that's very error prone. But also, it's very difficult to just redo the same query again. And these databases also get updated. And as they get updated, sometimes things get changed. So here, for example, I'm just showing the change of a gene name from HLA-DRA to MAMU DRA between ensemble releases 109 and 110. And the reason it's important that you can now do these queries computationally, so you can imagine you query these databases and you get information and you base your conclusions on that information. So it's extremely important to really understand how you got to the conclusions that you drew, given the information that you had at the time. So with GGET, you can now actually reproduce the exact analysis that you did 1, 2, or 3 years later. And also importantly, somebody else can do that as well. Okay, GGET has a website. Another thing that I found is really, really important as you write software for biologists or generally write software, documentation. So how does your tool work? How are people supposed to use your tool? And so this is kind of where all of GGET's documentation lives. You can scan the QR code and get to this website. And as you can see, GGET has really been very widely implemented. So this is a bit updated, actually. We just reached our 150,000th download between PyPy and Anaconda. And I think a huge part of that was actually the documentation that people was very clear how to use GGET and which problems it can solve. Okay, 2 very important things that I've already touched upon that I really wanted to emphasize when writing software for biologists. They may seem kind of obvious, but I see so much software coming out where this was not done. So I just wanted to emphasize it. So you can imagine as omics data, omics technologies explode, so do the tools that are being developed to analyze this type of data. So between September 2016 and January this year, 1,706 software tools were developed just for single-cell RNA sequencing data analysis. So that doesn't even take into account any other omics technologies. And here I'm plotting how often these tools were cited. And I'm just kind of using that as a very rough estimate for how much these tools are actually getting used. And you can see that what half of the tools are really just never used after they've been published. And the two things that I wanted to talk about are one, whether or not the tool is easy to install, and two, whether or not the tool gets updated. So here I'm just plotting the fraction of tools per kind of a bin of citation, and whether or not the tool is installable through PyPy or Bioconda, which are very widely used software distribution systems. And you can see that more highly cited tools do have a higher fraction in that they're PyPy or Bioconda installable. And similarly, tools that are being updated, presumably that keep up with dependencies, the developers make sure that the tools still run 1 or 2 years after they're first being published, also get cited more. So these are just very rough analyses trying to put numbers to things that I've just kind of also observed through experience being in this field. I think that tools that are not easy to install are just kind of dropped at that first hurdle of the user trying to use the tool. And yeah, especially as we get towards data that is just way too big to just throw into Excel as biologists and as software engineers, we really want to think about how can we put the two things together and accommodate people with less coding experience. Accessibility can come in many, many other ways than the few things that I touched upon today: documentation, installation, and keeping the tool updated. So another thing I wanted to mention, so this is a map of GGET website users by country. And we translated the website to Spanish and immediately noticed since a 35% increase in Spanish-speaking GGET website users. So that's another aspect of how software can be made more accessible. Okay, so I'm gonna stop here talking about GGET and kind of developing software. The developing software part will come back in the rest of my talk. But I guess, are there any questions right now about GGET and how all that came together and what it's being used for today? Okay. I don't see any questions. Okay. In that case, we're going to turn our gaze from the Earth to the sky, and I'm gonna start the virus part of my talk. And if any of you have seen this talk before, I do apologize. I do really love this illustration of some really big numbers. But this is a picture of the James Webb telescope taken by NASA. And this was the first picture that NASA released from the telescope. And at the time, this was the most detailed image of our distant universe. And according to NASA, this image with all of the stars within it is through the night sky, like holding a sandcorn at arm's length. If we were able to see the whole night sky at once, we would be able to count between 10 to the 22 and 10 to the 24 stars, which is the estimated number of stars in the universe. I'm going to use that number to put another number into perspective. And that's the number of individual viruses on Earth. So there's an estimated 10 to the 31 virions or individual viruses on Earth, which, by the way, also makes viruses the dominant biological entity on Earth. So that's like 100 million viruses for every star in our universe. So you can really see how viruses are just kind of ubiquitous all around us. I'm gonna today just focus on one realm of viruses within that, namely riboviria. There's many more riboviria than the 200,000 or so number that I'm showing here. This is just the number of riboviria that we have NCBI taxonomies for. And riboviria are special. Riboviria are RNA viruses, basically. And they're special because the vast majority of common human viral diseases are caused by viruses that fall into this realm of riboviria. So I think by now we've all heard a lot about coronaviruses, but also dengue, Ebola, hepatitis B, influenza, measles, polio. These are all RNA viruses or riboviria. And so it makes sense that we would want to look for these viruses in sequencing data. But in order to do that, using current workflows, like I showed in the schematic before, we get our RNA sequencing data. We have these text files of A's, T's, and G's. And the first step is to align that to a reference genome in order to figure out what the different RNA molecules were. This is the number of reference genomes that we have for riboviria, and these dots are to scale, by the way. So clearly there's a problem here. However, in 2021, a new database came out of viral reference proteins, namely the PalmDB. And I'm gonna tell you in a second what the PalmDB is, but basically the PalmDB contained almost 300,000 viral sequences. And this whole project started because Leo and I thought, wouldn't it be great if we could use the PalmDB as a reference when looking for RNA viruses in RNA sequencing data. Okay, so what is the PalmDB? So the PalmDB is a database of RDRP proteins, or RNA-dependent RNA polymerase proteins. It was put together by a collaboration between Robert Edgar and Arden Verbeyen. And basically what they did is they searched the entire sequence read archive, or SRA database that I told you about earlier, so for sequences that look like RDRP proteins. So that's 5.7 million ecologically and globally diverse samples. And they came up with a consensus database of RDRP proteins that cover all the RNA viruses that they found in all of these samples. And what this allows us to do is now we can build a reference for all of these viruses that is still very, very small and compact in size. So just to put some numbers to this, using the PalmDB, it covers an estimated up to 10 to the 12 virus species, and it's less than 600 megabytes in size. Versus if you wanted to build kind of a pan-genome, just putting all the NCBI viral genomes together, you would only have about the 6,000 or so viral sequences that are in the NCBI database, but the index will already be more than 100 gigabytes. So it would be very computationally expensive to try to use that to look for viruses in sequencing data. So I'm gonna zoom out again for a second and talk a little bit more about why do we want to look for viruses in sequencing data. So I've shown you before just by the sheer number of individual viruses on Earth, you can imagine that viruses are just all around us. And for example, the study found that there's a bunch of viral sequences that you can find just by sequencing blank laboratory reagents. And that's a problem because these contaminating viral sequences can lead to false correlations in research, but they can also make their way into healthcare, like what happened in the 1950s, where polio vaccines were contaminated with a carcinogenic virus. We're learning more and more that viruses might be the underlying cause in different cancers, but also neurological disorders. And right now, we're just really bad at keeping track of what the viruses around us, even those that can infect humans, are doing. I don't know if any of you heard of the bird flu spread in the US, but basically bird flu has been spreading undetected in US dairy cattle for months. And the only reason we discovered this is because it jumped to humans, at which point, of course, it was too late. So NASA had a mission called NEOWISE. They just terminated it, unfortunately. But NEOWISE was a mission that was keeping track of asteroids in our solar system and immediately flagging any asteroids that could potentially become dangerous to Earth in the future. So I think we need a viral NEOWISE to keep track of viruses around us. And actually, the lab that I just joined, the Sabeti lab at the Broad Institute of MIT and Harvard, is already working towards this goal, and they are setting up a global viral detection network called Sentinel, which is really amazing. So you guys should definitely check it out. Oh, I think there's a question in the chat. Oh, thank you. Okay. And yeah, again, if you have any questions, please just interrupt me. It is 11 p.m. over here, so also bear with me. Okay. So what tools do we have for a viral NEOWISE looking for viruses in sequencing data? There's a lot of different great tools out there that you can use to look for viruses in sequencing data. They all can use NCBI reference genomes as their reference. However, if we want to use the PalmDB, we need to be able to do something called translated search or translated alignment. And that is because the PalmDB is a protein database. So we need to be able to align nucleotide sequences to protein amino acid sequences. So that is called translated search. In addition to that, we want a tool that is able to retain single cell resolution and really make use of the power of all of the modern omics technologies that we have. So we need a tool that is able to do cell barcode and UMI tracking. And lastly, we want a tool that is accessible. So I've mentioned different aspects of accessibility. In this case, I am just looking at, is the tool easy to install and is documentation available? All right, so when we first had this idea of using the PalmDB to look for viruses in sequencing data, we realized there isn't really a tool that checks all of these. And so I set out to build that tool. I did that by building on the alignment tool Callisto. So Callisto was developed in the PAFTA lab to analyze single cell and bulk RNA sequencing data. And usually you have your nucleotide data, you align it to a nucleotide reference, and you get back a cell by gene count matrix, like the one I introduced earlier. In this case, we want to align our nucleotide data to the protein reference, in this case, the PalmDB. And by aligning to the PalmDB, instead of getting a cell by gene matrix, we get a cell by virus matrix that tells us for every cell which virus was in that cell. Whenever you want to align nucleotide data to amino acids using a nucleotide aligner, you can either reverse translate all of the amino acids into the nucleotide sequences, into all the possible nucleotide sequences, encoding that amino acid sequence, but then you again end up with a huge index because of the redundancy of the genetic code. Or you can translate both the amino acid sequences and the nucleotide sequences into a new non-redundant nucleotide code. So I don't want to talk too much about the algorithm, but there's one really cool fact about it that I wanted to mention. And that is the exact code that we use. So we did that. We translated both the nucleotide sequences and the amino acid sequences into a new non-redundant code so that we wouldn't have this explosion of the index due to redundancy of the genetic code. And the non-redundant code that we used to do that, so we basically made up our own new genetic code. And that was a comma-free code. And a comma-free code is basically a code that you can only read in the correct frame. So in the sequence on the left, if ACC encodes an amino acid and ACA also encodes an amino acid, then CCA and CAC cannot encode anything because you could get those by reading the correct words off frame. So that's a comma-free code because you don't need a comma to separate the words because it only makes sense if you read it in the correct frame anyways. And this is a mathematical theorem that you can solve. And if you solve this for a 4-letter alphabet, for example, A, C, T, and G, and 3-letter words, it turns out that you can make exactly 20 possible words, which happens to be the number of natural amino acids. And this was such a coincidence that in 1957, before the genetic code was decoded, Crick actually hypothesized that our genetic code, so he knew at the time already that there were 4 nucleotides and 20 amino acids. And so he hypothesized that our genetic code was a comma-free code because you get this magic number 20. He was wrong, but it turns out to work really well for our application here, where we need a non-redundant code that can only be read in the correct frame. So that's kind of what's happening under the hood. We are translating both the nucleotides and the amino acids into our new non-redundant code. And then we get back a cell by virus count matrix. And I just wanted to give credit here to Delaney Sullivan. So Delaney has written a completely new and better version of Callisto. And this translated search option is part of the new version of Callisto. So he was incredibly helpful in implementing the translated version and implementing all of this under the hood. So I just wanted to give a shout out to Delaney here. Okay. Talking about ease of use, this entire workflow can be run in 3 commands, including the installation. And that also includes masking of host sequences, which I'm going to talk about a little bit. I don't want to go on for too long, but I'll talk a little bit about host masking sequences. And so, yeah, basically, if you have single cell or bulk RNA sequencing data, you can, in just 3 commands, install the tools, generate your virus reference, mask host sequences, and then align your data. And in its most basic form, none of this exceeds the resources of a basic laptop. And if you were to run it on a basic laptop, you still get a speed of about 1.5 minutes per 1 million sequences. So this is also something, talking about accessibility, easy to use, do not exceed, in the most simple form, the computational resources that one would have on a laptop. Okay, so how do we do? So basically we benchmarked Callisto translated search against the ideal situation. If we did have a nucleotide genome for the virus, how would we do? And then also to Kraken, which is another tool that is able to do translated search, though Kraken is not able to retain single cell resolution. And across the board, we do better than Kraken. Also, so basically I'm mutating the sequences as we go from left to right here. And at high mutation rates, we do even better than the ideal scenario of having a nucleotide genome available. And so that's probably because if you align basically in the amino acid space, you are very robust to silent nucleotide mutations. So anything that doesn't change the actual amino acid sequence, we're still able to detect it. And then at the bottom, so Kraken and the standard workflow were only given the exact virus target sequence, but Callisto translated search had to not only detect the sequence as viral, it also had to assign it the correct virus. And so at the bottom, we're seeing the precision. And even as we're going into the very high mutation rates, we have very high precision for the viral sequences that are being detected. And then on the right, we're looking at some real data where there's a known virus. This is all SARS-CoV-2 data. And we're doing really well at detecting that as well. This one data point right here, where Callisto says there's no virus, but the qPCR says there is virus. You will not see that in the paper because I thought this is really weird. This has never happened that Callisto wasn't able to detect virus if there was such a high viral load by qPCR. And so I've asked our collaborator to rerun this specific sample, and it turns out they accidentally flipped the column of the positive control and the actual readout, and this is indeed a negative sample. So Callisto was sensitive enough here to detect an error in the actual metadata. Okay. So I wanted to just briefly mention post-masking. So if you're trying to detect virus in, for example, a human sample, you're going to have the problem that the vast majority of your RNA in your sample is going to be human and not viral. And so you have to be very good at separating human and viral RNA. And that's especially difficult because there's a lot of cross-species contamination in viral and microbial references. And this is something that happened this year, where the Salzburg group identified in another paper by Poor et al. It was published in Nature, but was actually retracted. Salzburg et al. Found that they had identified several sequences as bacterial when they were actually human due to this cross-species contamination. And for our workflows, we thought a lot about how do you properly mask viral versus host. And there's actually a lot of subtle decisions here that you can make that will lead to a completely different result. So I'm going to show you here an animation of 8 different masking workflows. I don't want to talk about the details too much, but the main thing I want you to take away from this is that there are subtle decisions that you can make in post-masking that are not necessarily right or wrong, but that are going to make a huge difference in your biological result. And so, we're really happy that we were able to describe these 8 different workflows. And I think here also documentation comes into play because we want to make sure that the biologists who are using our software understand the decisions that they are making that might influence the biological result of what comes out of the software. So one of the results that Salzburg was, so this is one of the results that Salzburg was showing. So on the left, we have the counts for different bacteria in the original study. And then on the right, the counts for the same bacteria that Salzburg found after extremely conservative post-filtering. But one of the things that I've noticed is that a lot of these bacterial genera are still present, even after extremely conservative post-filtering. And so if these bacteria are actually present in the sample, then why is it the correct thing to just throw out all of the sequences that also map to human? Because I think in reality, if these bacteria are actually present in the sample, we just don't know whether they're bacterial or human. And in our workflows, you do not throw out the sequences, you simply mark them as also in host. And that allows you to, instead of doing this very binary, we're going to throw out conservatively anything that even remotely masks to the host. It allows you to keep these reads in your analysis and later make a decision of how you want to weigh these reads if you want to revisit them. because what we find is if you do extremely conservative masking, you will throw out reads for confirmed viruses in these samples as well. So this is just one aspect of just a problem that we encountered that we didn't really anticipate, but then thought a lot about and really tried to solve this, or at least start to solve this as we were in the middle of this project. Okay. Yeah, so this is just to reiterate, instead of throwing away all of these reads very early in the analysis, and then you end up with really just a very small fraction of potential viral reads, which might also lead to false negatives. We keep all of these reads in the analysis and allow you to later make a decision on whether or not you have additional support to make the decision of whether or not those reads are actually viral or are actually a host. Okay, I'm going to very briefly mention some results of a data set that we analyzed. If I'm going too far over time, I can also skip some of this.
Yeah, maybe if you could rush through it.
Okay, I'll rush through it. Okay. Yeah, basically, we analyzed the data set of rhesus macaques that were infected with Ebola, and then their blood was sequenced over time. This data set also had a spike in, which that was important. So I'm gonna very briefly mention the spike in, but basically we recapitulate Ebola over time as well as in the correct cell types. So even though we're looking only for the RDAP, we still get all the correct trends, which is great. And then we also find a bunch of other viruses that are present, especially in neutrophils, probably because neutrophils pick up viral sequences through phagocytosis and pinocytosis. And I wanted to very briefly mention the spike in because, like I said, viral sequences are very ubiquitous. So we detected a lot of contaminating viral sequences, and the spike in allowed us to actually find these. So any cells that were in the spike in, as well as the macaque, were likely contaminating. And several of these, I've mentioned earlier that study where they looked at just blank sequencing reagents. We can find several of these viruses in those blank sequencing reagents, and we can trace those viral sequences in other studies that use those same sequencing reagents as well. So here's a completely unrelated study that came out earlier this year, where they introduced a new spatial RNA sequencing method, but they use the same sequencing reagents. And again, this viral sequence just pops up everywhere, clearly not confined to the boundaries of the tissue, so just clearly a contaminating sequence. So that was kind of cool. But macaques, if it's the same animal that was sequenced over time, especially within a short time window, they show the same virome, which is expected. We don't expect big changes in the virome if the time window is very short. And then the last thing we did in the paper is we show that for some of these viral sequences, in addition to Ebola, we showed that when the virus is present in the cell, there is a change in host gene expression. So we found some evidence that the presence of these viral sequences actually has an effect on that cell, indicating that that might be an ongoing viral infection with an unknown virus. Okay. Yeah, so now Callisto checks all of these columns. I'm gonna skip the future directions for now. Oops. No. My computer just, okay, great. Okay, and with that, I'd like to thank, like I said, this was mostly work that I did in the Pasta Lab in collaboration with a lot of members from the Pasta Lab. So I'd like to thank all of them and our funders, and also my new lab, the Sabeti Lab at the Grove. and I'll take all of your questions.